how do we then downturn or minimize the detrimental effects to cell danger response from things like pathogenic infection?
That's a very tough question to answer sometimes because it's really patient dependent in terms of what you try to find and what you try to dig through. So, as a clinician, we have to look at, what the patient is dealing with, have a right amount of clinical experience, and as was investigating, that's basically what it is. So we do a lot of functional lab and looking at these things. So, just kind of give you through what happens during the CDR response, that should actually explain how to actually address some of the things that I'm going to be talking about. And that's the use of phospholipids. And I love using phospholipids because our cell membrane is comprised of fat. And when you're dealing with the cell danger response and all these things that could actually damage the cell membrane, well, you can be needing these type of lipid therapy to add it into it. So when you're going through a cell danger response, for instance, a cell membrane gets depolarized, and then the mitochondria, right? We always think that mitochondria is about making energy. Well, that's not the case. Mitochondria is the brain of the entire cell, able to direct a immunological response. So instead of making energy, now it's actually going into a battle mode, right? And adding in additional immunological things to occur. So, forming a inflammatory fatty acid, followed by, to contain the overall damages within the cell, the mitochondria starts to pump out phospholipids to lock in the cell membrane. So this way there's no communication that goes on, is able to contain that damage inside and prevent further damages going on. And that's where the issue is. People are stuck in that mode, right? The cell's not able to communicate that well. And then when you are talking about poor epigenetic profile the patient might be dealing with, eating poorly, eating a lot of inflammatory oils, stress response, chronic inflammation that goes on, your phospholipid, your lipid level that your body's supposed to have, and we always know that fatty acid is very healthy, right? If that fatty acid level is in an imbalance or lacking because all the epigenetic profile has been piss poor, quote unquote, that's going to result in a lipid lacking in your body to help to contain all those damages that goes on. So there's a further cycling of cell danger response going on. And then the mitochondria is shock mode. They can't do anything to get you through. So in order to calm that cell danger response, what I do is I recommend patients to utilize phospholipids. So phospholipid complex, looking at phosphatidylcholine, inositol, serine, and ethylolamine, on top of adding in linoleic acid, because your cell is comprised of omega-6, omega-3 linoleic acid. And there's a lot of controversy beyond that, but you got to get the healthy linoleic acid of ALA to help to structure that inside the portion of the cell, along with some of the minerals, because obviously you need minerals to allow the cell membrane to communicate well, followed by some other additional fatty acids like butyrate to protect the DNA, to protect the cell membrane structure and allow the proper detox method and cells to communicate properly. So when you're looking at some of the further work that Dr. Robert Novia actually had done, he explains all these aspects in terms of what happens during a healthy response and a poor response in dealing with inflammation, result of the fact that the phospholipids are being depleted and the cell membrane is being damaged, cells not able to communicate, and a constant cyclic issue of cell danger response going on. And then the mitochondria is not able to orchestrate a proper healing as needed.
It's a really interesting take because most people associate mitochondria, the powerhouse, right? It's almost the muscle in that cell or something, when you're right, it's more of a brain, which is a powerhouse in its own and uses a lot of energy. But it really does direct a lot of the cell function. And then you talk about things like brain membrane potential and the idea of phospholipid therapy